DNA structure and organization. So if we were to look at these three organisms here and try to match their genome size, represented by how many of the individual monomers that are making up this double-stranded DNA, the base pairs. We have a salamander, we have an amoeba, and we have this Vitruvian guy at the bottom here. If you were to look at the size of their genome and take a look at the complexity of that particular organism, we'd come to some conclusions that would probably be incorrect. The salamander itself is the one that has 180 billion base pairs. However, it ends up with somewhere around, and these numbers jump around, but somewhere around 15,000 genes. The amoeba has a whole lot more base pairs than that, but much fewer genes. A human with relatively few base pairs, especially when compared to a salamander or an amoeba, has quite a few genes. And so we're going to look at the structure and the function of DNA, the pieces that make it up, but how it is built and how it is used are going to be two different things, as we can see here. So the units that are making it up are called nucleotides. They have three main components to them. We have a ribose sugar here, or a deoxyribose sugar, depending on which type of genetic material you're talking about. DNA, deoxyribose nucleic acid, or RNA, ribonucleic acid. We're going to be using one of those sugars, and it'll be that carbon there with the OH or not OH, just H, as to whether it is oxygenated or not deoxygenated, and that'll be your DNA. But three parts that make it up are going to be this ribose sugar, and again it could be deoxyribose, the nitrogenous base, which is up here, and there's going to be four varieties that we see for DNA that are going to be making these ones up. And of course we have this phosphate group, which we've seen as a chemical functional group on other materials as well. So a single nucleotide is going to have those three components to it. The bases themselves is where we get the variety of different components to make up the DNA. And so we have our purines, which are our adenosine and guanine, and we're just going to refer to them as A and G. And then we have our cytosine and thymine that are going to be making up our pyrimidines. Now DNA is going to have thymine in it. When we start talking about RNA, ribonucleic acid, it is going to replace that thymine with uracil. So here's a picture of them here, and as you can see, the nitrogen base is what is changing for each of these nucleotides here. And again, adenosine and guanine with a double-ringed nitrogen base, and thymine and cytosine with a single-ringed nitrogen base. So when DNA is together, and when these nucleotides are bonded to each other, they're going to be forming this chain that has the nitrogenous bases making up the rungs. So think of this as, and again, this is DNA here. We know that we have this double helix nature to it. We want to think of it as this ribose and phosphate is a component of it, which then has the nitrogenous base. So that is a single nucleotide there. And then it's going to be bonded to this other nucleotide here, which also has a ribose, a phosphate, and then it has the nitrogenous base pointing into the middle. And so each one of these ribose, phosphate with the nucleotide pointing in the middle, that is a single nucleotide. They all join together along this chain here, and this is called the sugar phosphate backbone, because it goes sugar, we got a sugar, and then we have a phosphate, joined to a sugar, joined to a phosphate, and so on and so on. And so it's a sugar phosphate backbone with the nitrogenous bases pointing into the middle, and that is what's making the rungs of this ladder. Now notice that one side of it is running parallel but in the opposite direction. And so we have the phosphate end up here. This is the sugar phosphate backbone of this last nucleotide, ends with a phosphate pointing upwards. And then down here you can see that this last sugar phosphate backbone, this last nucleotide here, has the sugar at the bottom here. And the matching side to it, the other side of the ladder, the other side of the double helix, is facing the other way. So it's almost as if this one's facing downward and this one's facing upward, if you consider the phosphate to be the upward end of it. And so we have a way of showing those. This phosphate is known as the 5' end, and the sugar is known as the 3' end. And so on one side of the double helix, we have our 3' end at the bottom, and we have our 5' end at the top. And on the other one that is parallel to it, it's the opposite. We have our 3' end up here, and then our 5' end down here. you could say that they are anti-parallel. They're in parallel with each other, but they're running in opposite directions. And again, it is the phosphate backbone. So you can see this part here is the phosphate backbone. And again, that is running in the opposite direction as the parallel one on the other side. And then you have these nucleotides that face each other into the middle here. This is why we call them base pairs, because those nucleotides are always paired up with one from the other side of the double helix. So the thymine and the adenine are going to be bonding together, so they're the ones that actually match up. They always match up with just that corresponding nitrogenous base on the other one. And remember, it's attached to, we're not showing here, the phosphate backbone. And again, we would have whichever direction this one's running, this one would be running in the opposite direction. You could look at it from either side, but the thymine is always going to be partnering with the adenine, and it is partnered with 2 hydrogen bonds. The cytosine and guanine are partnered with 3 hydrogen bonds. And so we see that we do form 3 hydrogen bonds, but often when we're showing them, we'd say, okay, this cytosine down here is just going to bond to a guanine here. We normally just show it with a little line like that, and we'd say the thymine above it is bonding with the adenine. So yes, this is forming 2 hydrogen bonds, and this cytosine guanine is forming 3 hydrogen bonds. We normally just show that with a single line showing the connection between them. Pyrimidines on the left here, purines on the right. So each of these sugar phosphate backbones, again, a little bit easier to see here, are running anti-parallel. So whichever direction one of them is running, so see these sugar phosphates are facing upwards with the phosphates being on this end, and that is the 5' end. And that means on the other side, the phosphates are going to be pointing downward here. So this is our sugar phosphate nucleotide here, and that means this end is the 5' end. They're anti-parallel to each other. This is going to be really important when we get into talking about how we copy these strands, because when we separate this double helix, we're going to be looking at each of these 2 different strands. We're going to break these hydrogen bonds in the middle, and since they're running anti-parallel, the way in which they're copied is going to have to be adjusted for accordingly. Eukaryotic DNA, a little bit more complex in terms of how it's organized, and then we'll briefly talk about prokaryotic DNA. So in order to fit the amount of DNA, we've got about almost 2 meters of DNA inside each of these cells, if we're talking humans. Very, very narrow, but quite long, and we've got to fit this into the nucleus. It has to be packaged up quite well. So what's going to happen is this normal double-stranded DNA, that's how we sort of picture it when we're imagining DNA, but it's going to have to be able to get packaged up in a way that can condense it into the nucleus, especially when we're going to be doing things like mitosis with it. And so these lengths of DNA are going to be packaged around these proteins called histones, and they form this nucleosome. So you imagine the DNA being sort of wrapped around and forming like a little bead. That's going to be these nucleosomes. The nucleosomes are then going to be coiled into these chromatin fibers. And so you can imagine these sort of beads get all sort of stacked up together. And then that is going to be further condensed. And eventually we get, after enough packaging, we end up with what we know when we did our studies on mitosis, these chromosomes. So again, the double helix DNA has to be packaged quite elaborately in order to get this condensed version of DNA that is the chromosomes. Prokaryotic DNA, a little bit simpler. The bulk of their DNA is found in this nucleoid. Remember, they don't have the nucleus, the organelles that eukaryotes have, but they do have this nuclear region called the nucleoid, and it tends to be supercoiled. And so you can imagine this. You have this coiled DNA, and just like you can take a coil and then further twist up that coil. That's what a supercoil would be. If it is double-stranded, it could be just the double-strand DNA, or it could actually form a loop. So you have this circle of it, but there are prokaryotes also that have single-stranded DNA, and so they could be just a linear strand, or they could also be in the form of a loop. And this is their found in the DNA that's found in the nucleoid region of the prokaryotes. Because there is this other DNA that is non-nucleoid. So nucleoid is sort of like their main functional DNA that they need to be alive. But they also have this extra genetic information called plasmids. And these are basically sort of extra DNA that can be picked up from their from other bacteria or from their environment, and a huge tool that we use in genetic engineering in order to get certain genes into these bacteria. And essentially what they are is this circle of DNA, which is got all these different codes. So you can see here's a map of a particular plasmid here, and we have these different genes on it. The bacteria can pick up this extra DNA, and they can use it to start expressing and making things that they couldn't make before, as long as those things are coded for in this plasmid. Not a good thing on it, it would be something like bacterial antibiotic resistance, because they can actually pick up that DNA from another bacteria that has dropped its plasmid into the environment, maybe destroyed it or whatever, but they can actually pick up that plasmid from its environment, and then it will now have those abilities. Telomeres, last note on this is that DNA, and so we're going to be looking at eukaryotic DNA replication, but during replication, the 5', 3' nature of the DNA means that there's going to be this problem that happens at the ends of the chromosomes. So there's going to be this one side, because remember they run in opposite sides. They've got this 5' over here, and so that means the 3' end is going to be over here, and then the other one is anti-parallel to that, so it's got the 5' on the right here. And so when this DNA gets copied, there is one end that is going to be left and it's unable to be copied. So essentially each time the DNA gets copied, a little bit of the chromosome on the end cannot be copied because of the way that it is copied. More on that later. And this causes that every time it gets copied, the DNA actually gets shorter and shorter and shorter. So this would obviously be a problem if each time you replicate your cells, you have less information in them. And so the cells deal with this with these things called telomeres. So each time it gets replicated, they would lose some information. And so at the ends of the chromosomes, there are these non-coding regions that are called telomeres. And so as the DNA gets copied, we lose that piece, and the next time it gets copied, you lose another piece, and the next time it gets copied, you lose another piece. They get shorter and shorter. So as long as the ends of the DNA are not really important parts of the DNA itself, but sort of like non-coding regions, not important for making proteins, if you lose that pieces of information, it's not such a big deal. However, obviously, that can only go on for so long before you start eating into some important information.